Good morning to all of you. Today I am going to discuss about agronomic traits from biotechnology. Just to create a transgenic plants to protect the plant from different insect, different pests, different virus, different bacteria, and different animals. That is the main motto. And the production of quality food that contain huge quantity of nutrient like vitamins, minerals, carbohydrates, protein, amino acids, and plant can survive even in the scarcity of water in high alkaline soil. So this is the objective of uh, uh, production of transgenic crop regarding different traits like herbicide tolerance plant, that is the transgenic plants, insect resistance plant. Herbicide resistance or tolerance plant, that is a variety of uh, soybean. That is a variety of soybean. Okay, so that is the variety name is round up ready. You know what is the source of protein in the agriculture? That is the soybean is the most potent source of protein.
Yes, sir.
Yes, not only protein, soybean oil. You can get the oil also from the soybean. So generally, weed. Do you know weed? Unwanted plants or unwanted crops besides main crop that decrease the production of the main crop. That is known as weed. So just to check the growth of weeds, unwanted plants in the crop field with the help of herbicide chemicals. So when we are spraying the herbicide, that herbicide do not recognize whether that is the crop plant or weed plant. So scientists have developed a variety of crops of soybean crop, so they can in turn transfer the gene regarding the tolerance of herbicide. Means if you are spraying the herbicide chemical to kill the weeds, the plant is not affected. The transgenic plant will not affected by that weed. That is the concept. So only the herbicide, that the chemical will kill only the weeds. That is the concept. So scientists have transferred the gene inside the chloroplast of the soybean plant, and against the herbicide name is the roundup, that is also known as glyphosate. It is a chemical that is herbicide. So generally farmer is using that glyphosate or roundup, that is the trade name. And scientists have developed a variety. The variety name is roundup ready. The variety name is roundup ready. So this is the concept. This is a model for production of herbicide tolerance plant against the herbicide chemicals. So lot of transgenic plant have developed against the herbicide tolerance. That is the gene that is responsible for not affecting the uh, effect of herbicide, means detoxify the effect of that chemicals. Because when the weed is growing in the field, you cannot separate this part will be the for weed and this part will be for crops. So generally along with the main crop there is a weed is present, and it is not selective. So chemical will never recognize whether it is the crop plant or wheat plant. So that's why scientists have developed such type of gene inside the crop, so that there is no effect of that chemicals on that crop, and then the plant can grow quickly and produce huge quantity of soybean seed for getting soybean chunk or soybean products. So you get different type of food product, soybean cheese, soybean paneer, like that, soybean oil. from the soybean plant. Next, that is the creation of transgenic plant against the insect. What is BT cotton or BT brinjal? Means BT gene is going to transfer inside the plant. What is the source of BT? That is the BT gene is present in bacteria, that is present in soil. Where is present the BT gene? Inside the bacteria. That bacteria is present in soil. Now, after isolating the BT gene from bacteria, where it is present? Inside the plasmid of that bacteria, Bacillus thuringiensis bacteria. So, scientists have isolated the gene responsible for production of BT crystal protein, and transfer in the plant. Which plant? Inside the cotton plant, inside the brinjal plant. So that if they get attacked by insect, what are the insect? Cotton bollworm for the cotton, that is going to degrade the cotton ball fiber of inside the cotton ball, or the fruit of brinjal, that is fruit borer. If you observe in your market, vegetable market, there is so many worm are there, uh, the insect are there inside the fruit of the brinjal. So that is going to destroy the quality of that brinjal and degrade the whole food edible part of the fruit of brinjal. So if the gene against the insect resistant is there, then there is no chance of affecting the fruit of the brinjal or ball of the cotton. How? When it will, that insect will suck the juice of that plant, there is a formation of crystal, because there is a BT gene in that plant, and BT gene is going to release the BT crystal protein. What happens? This is generally present in soluble form in the plant, but when the insect is sucking, when it will go inside the crop of the insect, then it becomes solid crystal, and that solid crystal act like a blade, and it will rupture the intestine wall and thus kill the insect. So this is the concept. So this is the area of research to create a transgenic insect, uh, transgenic plant against the insect, like cotton bollworm or foot or shoot borer, Econostic mesenteroids. That is the name of scientific name of foot or shoot borer. Next is disease resistance. Scientists have developed so many plants, that is transgenic plant that contain the gene responsible for disease resistance against different type of disease, like a viral disease, like bacterial disease. You will find curly type of leaf in case of papaya, curly type of leaf in case of so many plants. So just to show the curling of the leaf, that indicate the presence of virus in that plant. So that variety, the scientists have developed the variety so that they know effect of virus, they know effect of that bacteria. So this is the concept. Next is abiotic stress. What do you mean by abiotic stress? Means a scarcity of water, a scorchy sun temperature, abundance of salt in soil, abundance of metallic compound in the soil, that affect the growth of the plants. So if there is scarcity of water, plant will not survive. So scientists have developed the gene DR, drought resistant gene, and transfer in the wheat plant, because wheat plant require water. So where that gene is present? In the jowar, bajra, that is growing in the area where there is a scarcity of water. So such type of gene is present in that variety of plant, and we can transfer the gene responsible for drought resistance. So that is going to transfer in the main crop like wheat. So that thus we can grow huge quantity, produce huge quantity of wheat grain in area where there is a scarcity of water. Generally that area is the area of arid region, that is the rainfall, there is a scanty rainfall. So this is the concept for transferring the gene after isolating the gene from the crop, which can survive even at very low water or a scarcity of water. So osmolytes, so many osmolytes are synthesized by the plants of such type of crop. In case of jowar or bajra, like mannitol, sorbitols. So these are the osmolytes. So that type of osmolytes that is secreted by after action of particular genes. So what is the name of that chemicals that can be osmolytes? So that can help for tolerating the plants even at a scarcity of water. So this is the concept of stress tolerance. Okay. So just like so many compound like butane. Okay. Then mannitol, trihalols, fructans. So these are the butane. It is also osmolytes. Okay. Then fructans. It is also osmolytes. Trihalols. It is also a carbohydrate, that is osmolytes. So when these compounds are secreted by the plant, even in a scarcity of water, plant can survive. So can you learn these things? Osmolytes. It is a chemical that is secreted by plant that is coded by particular gene in any other variety of crop, not in the commercial variety. So first you isolate the gene responsible for production of the osmolytes like trehalose, like mannitols, like fructans, like other chemicals, butane. So these are the chemicals that is responsible for maintain the absorbing, maintain the moisture inside the plant, and plant can survive even at a scarcity of water. So transferring the gene that is responsible for secretion of the osmolytes in the commercial variety, and then plant can survive even in a scarcity of water, even at high temperature, even at high saline soil condition, even at the soil where there is a high metal compound. That is adverse situation. So we are going to create a transgenic crop against a biotic stress. We can also, yes, yes. So we can also, yes. Uh, do you want to just uh, you can use these things? Yes. So just see the gene is responsible for each type of compound production, like osmolyte, like mannitol, sorbitol, that is the alcohol, sugar alcohol, or butane, that is the amino acid or protein, and trehalose, it is also a carbohydrate. So that is the different type of chemicals that is coded by particular gene, and that gene is not present in all plants, only present in selected wild variety of plant. So you isolate the gene and transfer the gene in the plant of a commercial crop. What is the commercial crop? That is the wheat, that is the rice. So just to maintain the moisture content, the osmolyte gene is essential. Only then plant can grow even in a scarcity of water, even at high temperature, even at saline soil, even at the soil have high metals. So this is the concept. Okay. So try to know this is the abiotic stress. This is the abiotic stress. Next is value added traits like oil. You know, oil. You are going to increase the unsaturated fat. If you are comparing the saturated fat and unsaturated fat, then the chemical that create allergy, some chemical that is synthesized the plant, that create allergy, just to reduce the allergic compound, reduce the saturated fatty acid, enhance the unsaturated fatty acid, then enhance the vitamin contents in the plant, like wheat, like rice. So bt, just golden iron rice, it is example of the plant that contains huge quantity of transgenic plant that contain huge quantity of what, that is the vitamin A, that is beta carotene. So scientists have developed the Ingo Portugas, a scientist Ingo, INGO, Ingo Portigas, Portugas has developed the golden iron rice. What is the meaning of golden iron rice? That contain huge quantity of vitamin A, that is beta carotene, that is the precursor of vitamin A, and iron. So generally the poor people are not cannot afford the buying the carrot, buying the apple, because it is the source of vitamin A, or buying the papaya, but they can purchase the rice. So if there is supplement of that vitamin in rice only, then poor man people problem can be solved automatically. What is the problems? Problem is that due to deficiency of vitamin A, there is a disease. What a type of disease is there? What type of, yes? What type of
Sir,
disease is there?
night blindness.
Light night blindness. This is the concept. So the night blindness, it is a disease that is caused. Okay. So night blindness, it is a disease that is caused due to deficiency of vitamin A. Next disease, that is due to iron deficiency. Okay. So what type of disease they are due to what type of disease caused due to iron deficiency? That is anemia.
Sir,
So
yes, sir.
yes, anemia. It is vitamin B12, that is cyanocobalamin, that is activate for the production of hemoglobin. So generally, what happens if there is a deficiency of vitamin B12? Automatically, what iron will not absorb in your body. If there is a deficiency of vitamin B12, automatically iron will not absorb in your body. And if iron will not absorb in your body, what happens? There is no formation of RBC, erythropoiesis. So this such things means if the iron content is there in your food, and if there is a deficiency of vitamin B12, in that situation there is no formation of RBC. So if there is a sufficient amount of vitamin B12 and folic acid, that is iron, only then there is a formation of RBC, that is hemoglobin. Okay, so golden iron rice is the option transgenic crop, that is the option to fulfill the demand of uh, uh, that is the vitamin A, deficiency of vitamin A and vitamin iron. So scientists have developed the transgenic crop, that is golden iron rice, and also developed the transgenic crop again the different minerals, important minerals, that is inside the pulse crop. Okay, and amino acid in case of cereal crops, and in case of pulse crop. So these are the concern that creation of transgenic plants against production of unsaturated fatty acid. Why unsaturated? Not why not saturated? Because unsaturated fatty acid do not cause any cardiac problems. You know, saturated fatty acid cause arteriosclerosis, hardening of arteries, and sometimes heart attack. So if there is a huge quantity of unsaturated fatty acid, huge quantity of that is the omega fatty acid. You know, omega fatty acid generally present in almonds, in the walnuts. So these are the source of omega fatty acid. So if you are going to enhance that type of fatty acid in the plant, then it will not affect your heart will be normal. Okay, so that never cause heart attack. So this is the concept to enhance the unsaturated fatty acid. Okay, and it will never cause arteriosclerosis disease. So scientists have developed such type of transgenic plant that do not count any allergic compound, just to reduce the compound allergic compound in the plant. So these are the area of research where scientists have developed the different transgenic crops. Next, the nutritional quality like protein, fat, carbohydrate, shelf life. Have you heard about flavor saver variety? Flavor saver, it is a variety of tomato that due to formed by antisense RNA technology. Antisense RNA technology. You remember these things? A variety of tomato that is formed by antisense RNA technology, means there is a delay ripening of the fruit. Why? Because there is no formation of ethylene inside the fruit as soon as possible after plucking the tomato from the tomato fruit from the plant. So why? Because there is blocking of the pathway that is responsible for ethylene production. Why I am telling ethylene production? Why I am telling ethylene production? Because ethylene is responsible for ripening of the fruit. So at the gene level, scientists have blocked the formation of the ethylene. So that's the way, that is the antisense construction of antisense RNA to block the function of the gene responsible for production of ethylene. Means some enzyme is responsible for production of ethylene. So that enzyme is coded by a particular gene, and that if you are going to block the particular gene, then there is no ethylene production. If there is no ethylene production, there is no ripening of fruit occur quickly. So you can sell that tomato variety from Punjab to all over the country, from Punjab to America, from Punjab to Japan, from Punjab to other part of the world. So this is the concept, from Chandigarh to all the part of the world. So that is the flavor saver variety, and you can also enhance the flavor aroma in the plant variety through gene transfer inside that plant. So this is the concept. You can develop the aroma. So these are the different fragrance. Means if you are going to transfer the gene from basmati rice to non basmati rice, then the non basmati rice is also appear like basmati rice due to presence of fragrance, the gene responsible for fragrance. Then allergen reduction, I have already discussed the so many chemicals like boa. You know boa, b capital BOAA, and that is the chemical that cause allergy, that cause a disease, that paralysis in the pulse crop. So scientists have developed the variety that do not contain boron. Okay, so that is the concept for reducing the compound that cause allergy. And nitrogen fixation, one of the most important aspect in the field of biotechnology, means there are some plants that is able to fix nitrogen, that is known as leguminous plant. Okay, you have read these things in class 7 or 8. Okay, so nitrogen fixation is only done by some group of plant that is known as leguminous plant. But scientists, they have developing they are developing the variety of the plant, although it is not a leguminous plant, but transfer the NIF gene, NIF, NIF gene and NOD gene. So NIF gene and NOD gene responsible for nitrogen fixation, nodulation, root nodule formation. Okay, so when there is a formation of root nodule, automatically bacteria that is responsible for nitrogen fixation will come and will get the host shelter for nitrogen fixation, and then automatically fix the atmospheric nitrogen. This is the concept. Means conversion of non-leguminous crop to leguminous. How? Means if it is if any crop has ability to fix nitrogen, then it is come in the category of leguminous crop. Then yield, that is the very important traits, that you have to produce huge quantity of food grain, because population of India, population of world is growing geometrically, exponentially. What do you mean by geometrically or exponentially? 2 per power n. Okay, so if n is the 1, then 2. If n is 4, how much? 16. Not like 1 2 3 4 like increasing population, not like that. Means exponentially it is growing. So how can you overcome the problem, food problem? Through creation of transgenic plant. Means this is the great challenge for scientists to fulfill the demand of food of the growing population. So how it is possible? Through creation of transgenic crop to improve the quality. Okay, so this is the major problem and faced by the scientists in the field of agriculture. So there is a lot of opportunity for producing such type of variety that produce huge quantity of food. Okay, through transgenic plants. So this is the concept. Next is pharmaceutical production, vaccine. I have already discussed this thing, vaccine production in inside the plants, inside the, and that is known as molecular farming. Then production of useful protein. That protein can act as hormone. That protein can act as a antibiotics inside the plants. That is the concept. That is production of pharmaceutical. That is the concept. Means molecular farming, one of the major area of the research. You are going to create a transgenic plant for production of vaccine, for production of edible. That is the concept of edible vaccine. So that is the production of any useful protein that can be used for treatment of different type of disease. So these are the different traits you have to consider in the field of agriculture biotechnology and the plant biotechnology. So this is the important criteria you have to consider when you are doing any research. So this is the meaning of agronomic traits from biotechnology, mainly for plant biotechnology. Okay, समझ में आ रहा है कुछ? Are you listening my sound?
Yes,
हाँ,
sir.
तो समझ में आ रहा है कुछ क्या मैं बोल रहा हूँ कि प्लांट बायोटेक्नोलॉजी या एग्रीकल्चर बायोटेक्नोलॉजी में आपको ट्रांसजेनिक प्लांट बनाना। ट्रांसजेनिक प्लांट आप बनाएंगे कहाँ, कैसे? यू सेलेक्ट द जीन वेयर इट इज प्रेजेंट इन वाइल्ड वेराइटी। वाइल्ड वेराइटी मीन्स those plant variety from which you are not getting huge quantity of food, but that contain the quality gene that is going to protect from different type of insect, different type of microbes, different type of pathogen. So we can isolate first and then transfer in the main crop, that is the commercial variety from where we are getting the food. This is the concept. Means GM crop. What do you mean by GM crop? Other name of transgenic crop, that is genetic modified. How can you do the genetic modified? First you isolate the gene from the wild variety of the crop, same crop, and then transfer the gene after isolating the gene from the wild variety into the main crop, and that crop now become genetic modified crop. Means you are going to change, alter the gene. Alter means adding the gene. Okay, so this is the concept. Now, you know what do you mean by sustainable development? Means you are going to maintain the production of the crop. Means you cannot stop the population growth. So can you reduce the production of agriculture crops, food? No. Why? Because there will be a scarcity. If there is a scarcity for food, for growing population, hue and cry स्थिति will situation will be created. Hue and cry, अफरा तफरी मच जाएगा, पता है ना? So just to fulfill the demand of growing population, okay, so 10 billion people expecting to inhabited on the earth by 2050, 10 billion people. So just to maintain, if you are not going to produce more, you have to maintain the amount of production of the food grain. So that is known, that is the concept of sustainable development. Means you are not going to reduce the production. How you are going to maintain the production? Through creation of transgenic crop, to change the gene. Why? Because in due course of time there is so many infection from different type of bacteria, fungus, virus occur by uh, uh, environment on the crop. You have heard the news tidda, that is going to destroy the whole crops in major area of Rajasthan, Madhya Pradesh, Uttar Pradesh, that is coming from Pakistan. Okay, so I think you have heard in the G News or in Aaj Tak. So that is the concept. Means you have to protect the plant. Why? Because this is the only source for food for all the people. So you have to change the genetic composition and create a transgenic crop. And when that insect will attack the crop, due to bitter taste of the juice content present in that crop, it will repel. It will go away. So this is the concept you have to develop. Means you have to develop such type of variety of crop that it will produce a good quality of food grain. But when it will attack on the leaf or a stem part after sucking the juice, the juice content have such type of compound that it will kill that insect. So this is the concept of sustainable development. Means sustainable development is the development that meet the need of present without compromising the ability of future generation to meet their own need. Compromising the future generation means you cannot disturb the food chain and food web, but you have to create a transgenic crop and produce the huge quantity of food grain to fulfill the demand of present and coming future generation. So you have to extend up to such a way your agriculture biotechnology or plant biotechnology, so that a variety of transgenic crop can be developed from different for different crops. So this is the target, that is the motto of sustainable development. Now biotechnology in India, you know, it was started the department of biotechnology in the form of department of biotechnology in government of India. So it was started in 1980. Okay, and up to Vision 220 documents have been prepared by Technology Mission Forecasting and Assessment Council. Okay, so this is the organization of government of India, TIFAC. Vision 220 document has been prepared by Technology Info T for Technology, I for Information, and F for Forecasting and Assessment Council. So that is going to assess the production of present food grain production and future food grain production after estimating the population in near future. So how much production scientists will produce through creation of transgenic crops. So that is the motto of that TIFAC, who also include the biotechnology. Indigenously developed hepatitis B vaccine was developed by Santa Biotech hepatitis B vaccine. Okay, so in 1998, Santa Biotech in India. So there are nearly 30 company have developed to development of modern biotechnology in India. 13. So you know Kiran Majumdar is she the leading lady in the field of biotechnology. Kiran Majumdar. If you will join in the company of Kiran Majumdar, your monthly salary will be 4 lakh or 5. Okay, in India, that lab is present in Bangalore. Kiran Majumdar. She is a great industrialist in the field of biotechnology. So if you will join in that industry, your monthly salary will be minimum 4 to 5 lakh. Okay, so there is lot of opportunity. 30 or more than 30 different company are there in the field of pharmaceutical industry, in the field of agriculture, in the field of medical science. So lot of opportunity are there in the biotechnology. Currently, India sharing the global market value is just 1.4%, 1.5%, and that is the 10 year before data. Now I think it is up to 5%, and this value is up to dollar 1.05 million. Now there is a high growth rate in the field of pharma industry, or in the field of medical industry, for production of different instrument in the field of medical science, and then production of different drugs, medicine, and then the field of agriculture, transgenic crops. For they are getting 32% high growth rate. India is positioned to become a major layer in the coming years. So India will lead in the field of biotechnology also, not only in information science, information technology. India will be leader in the field of biotechnology also. So many scientists are working in foreign country. The brains are working in the foreign country like Japan, like America, USA, in the field of biotechnology. Indians are there, and they are doing better research, and they are getting so many name and fame. So I think you have heard about the father of green revolution in India. So MS Swaminathan. So these are the leader in the field of agriculture and plant biotechnology. for producing huge quantity of food grain. Now, if you see in the global scenario, there is a major leading country is USA. After that, China, then Germany, then France, then so they are the leading country in the field of biotechnology, pharmaceutical industry. Okay, so you see the growth rate that is 16% and crossing the production of 100 millions, 100 billions, not millions dollars by the year 2010. This is the 10-year before data. And US promote the biotechnology, and they have developed the uh, taximati, you know, basmati, it is a rice variety, and they have tried to patent the taximati. This is the indigenous American origin. They are claiming that they are in American origin, means basmati is American origin. They are also claiming that the neem plant is also American origin, but India have fight um, in the international court and had defeated in the regarding basmati, regarding neem plant. There are so many useful compound present in neem, and then haldi, tamarind, so that have capacity to fight against cancer. Okay, so such type of
सर
possibilities. Yes, बताओ.
सर पहले अंग्रेज लूट के चले गए।
बोलो. अंग्रेज
सर
भी
पहले
लेके,
अंग्रेज
यस, अंग्रेज भी लेके गया, लेकिन अभी मैं प्रेजेंट सिनेरियो की बात कर रहा हूँ. मैं प्रेजेंट सिनेरियो की बात कर रहा हूँ कि बासमती जो राइस है, हैव यू हर्ड अबाउट बासमती सेंटेड राइस?
ये तो मैं
हाँ, द सेंटेड राइस जो बासमती है, वो अमेरिका चाह रहा था कि ये नहीं, ये हमारे टैक्सीमती एरिया है, वहां पे ग्रो होता है, जबकि उन्होंने यहाँ से सीड ले गया और वहां ग्रो कराने लगा. लेकिन this is the Indian origin, only in India. It was first time developed in India only. So they have fight that the patent will be for America only, but India has filed a petition regarding that and have won that claim in the international court. They have also trying to adopt the neem plant, okay, and the product of the neem plant. There is large number of products are developed from the neem plants, and they have also tried to claim the uh, that is haldi, tamarind. Haldi, जो जानते हो ना, haldi, that is beneficial, that enhance the immunity. So this compound, that neem compound, neem plant में जो होता है ना नीम प्लांट, जिसको मार्गो भी बोलते हैं, उसमें सो मेनी कंपाउंड एजाडायरेक्टिन होता है, जो कि इम्यूनिटी बढ़ाता है, दैट प्रिवेंट द ग्रोथ ऑफ बैक्टीरिया, फंगस, वायरस, इंसेक्ट, एंड एंड द you will find the haldi. There is so many useful compound that is present in haldi, uh, that have ability to fight against cancer. So they are trying to adopt that thing, but they cannot do the patent. अगर उन्होंने पेटेंट एक बार कर लिया तो उसके परमिशन के बिना आप कुछ बना ही नहीं सकते हो. किससे? नीम प्लांट से. किससे? आप हल्दी से. Because patent is in the Indian name, so you can produce any compound, any medicine from the neem, from the haldi. This is the concept. अगर आपने एक बार पेटेंट बना लिया, जैसे आपने बताया था ना कि पेटेंट किया है इंडिया एक केमिकल, एक ड्रग्स का, तो होल वर्ल्ड इज आस्किंग टेकिंग परमिशन, टेकिंग दैट मेडिसिन इज ओनली सेल बाय इंडिया, not other country, because that is the patent is given by the name of India only. So I think you have heard this in this genius कि एक मेडिसिन था अगेंस्ट ट्रीटमेंट ऑफ कोरोना वायरस, जो कि अमेरिका भी चाह रहा था, फ्रांस भी चाह रहा था, जर्मनी भी चाह रहा था. तो why they are asking that provide me medicine, because that medicine was patent only by India. That's why, this is the reason. यही है, यही मैं बोल रहा हूँ. तो सो मेनी रिसर्च है इज गोइंग ऑन इंडिया रिगार्डिंग द ड्रग इंडस्ट्री, एंड द ड्रग इंडस्ट्री इज टोटली बेस्ड ऑन बायोटेक्नोलॉजी. CMAP, lot of organization are there like CMAP, then Central Drug, CDRI, Central Drug Research Institute, then NIPER in Mohali also. So that is the national institute, any international institute, that is ICGB, International Engineering for Genetic Engineering, International Center for Genetic Engineering, Delhi. And there are only two international genetic research institutes. That is, one is India, that is Delhi, and one is Italy. Okay, so that is the um, Rome. So these are the two
सर
centers.
टाइम हो गया
हाँ ठीक है, कोई बात नहीं. So that is the concept in the present scenario. NIH is observing and responsible for production of different chemical compound and give the patents, give the authorization, and National Institute of Health. Okay, and National Institute of Health is an international organization. ठीक है ना. तो आपको समझना चाहिए, कुछ भी आपको पब्लिकेशन बनाना है या प्रोड्यूस करना है तो NIH में NCBI, नेशनल सेंटर फॉर बायोटेक्नोलॉजी इंफॉर्मेशन में भेजना पड़ेगा. Then you will get the authentication. So this is the global scenario, and you try to know the what are the different traits in the field of agriculture biotechnology and plant biotechnology to produce the quality food grain, to produce the quantity food grain to fulfill the demand of growing population. Okay, so all of you must go through the book. Okay, और आपको तब पता चलेगा ये इसमें क्या लिखा हुआ है. कुछ कुछ जो टर्मिनोलॉजी आपको जानना है, जैसे डीबीटी, इंडिया का डिपार्टमेंट ऑफ बायोटेक्नोलॉजी, उसमें बहुत सारी इंफॉर्मेशन है जो कि आपको डिफरेंट व्यू देगा कि आपको क्या करना चाहिए. ओके, थैंक यू.